Hi, my name is Madison Gerdes, and this is an update on my thesis research titled Saltwater Intrusion Drives Ghost Forest Formation and the Loss of Old Growth Bald Cypress in the Northeast Cape Fear Watershed. This past spring of 2025, I received the Aaron K. Rice Applied Learning Award, which supported this work over the past semester, and without which much of this project would not have been possible. Starting off with some background information, we know that ghost forests or forests of standing dead trees are emerging along the US mid-Atlantic coast from relative sea level rise, which brings salty ocean water further into our freshwater wetlands and up our rivers. This sea level rise, driven by anthropogenic global warming, can also be driven by natural processes such as land subsidence. On this map that you see here on the slide by the USDA, we can see extensive ghost forests mapped along the Chesapeake Bay and around Florida. But zooming in closer to North Carolina, we see that there have been extensive ghost forests recorded along the Albemarle and Pamlico Sound. But the true extent of the ghost forests within Wilmington have not been mapped at this national level. We only see light and moderate ghost forests reported along the lower Cape Fear River out by Southport and Oak Island. And here in Wilmington, sea level rise is also exacerbated by dredging of the Cape Fear River for navigational purposes, which allows for further saltwater intrusion to occur, with the tides mimicking the effects of sea level rise by bringing more salty ocean water upriver through that now deeper channel that they dredged. Here in Wilmington, all of this combined creates a scene that looks a lot like this. This was taken here in Wilmington. Um, but as I mentioned previously, this is not what we're seeing mapped on a national scale. Now, we know that dredging is exacerbating this problem because when the river is dredged, we see a response in the tree ring record. This here is a plot of suppression events at Smith Creek, which is a tributary of the Cape Fear River, in response to dredge events, which are denoted by those red arrows with their depths. So, past the 1940s, we see a fairly clear relationship between dredge event and tree response. But what about in the 1930s when we see no response to dredge events, or in the 1860s when we see suppression before the dredging began? So, to untangle this, we need longer records to see what was going on further and further back in the past before dredging to uncover how trees have historically responded to other suppression events. And the need for longer records takes us to the bald cypress tree. So, bald cypress are a forested freshwater wetland tree found throughout the coastlines and the rivers of the US Southeast. They are a keystone species for their local environments, and they are extremely long-lived as the fifth oldest tree species currently identified in the world. They provide extensive ecosystem services, including flood control, carbon sequestration, and they are incredibly sensitive to salinity, tolerating only 2 parts per thousand. So the problem then becomes that because they only tolerate low levels of salt, they are dying and forming ghost forests as the salt water intrudes. Further, it's been found that the older bald cypress are less resilient to saltwater intrusion than their younger counterparts, and so the old growth keystone trees which have the fuller and longer records, are being lost first to dredging and sea level rise. So, my project aims to answer the question of whether the current suppression that we're seeing is unprecedented in their full dendrochronological record. And is this driving accelerated ghost forest formation that we're currently seeing to a degree in which the ecosystems cannot recover from? So, to answer this, over the past semester, I investigated the Smith Creek tributary of the Cape Fear River, which you can see here south of the Wilmington Airport. We have two sites, one in the high salinity zone near the 23rd Street Bridge, which you can see in box A, and then another in the low salinity area near the North Kerr Bridge in box B. And we see a very clear salinity gradient between these two sites, which can be seen in photos, which I'll show you in a moment, as well as the tree ring record. So this is box A, the high salinity site. As you can see here in the inset photo, we have extensive standing dead trees and generally younger trees compared to the low salinity site. And this aligns with the hypothesis that older trees are dying first as the saltwater intrudes. So in the figure, you see red points, which are dead trees. This is by no means all of the dead trees. This is just what was sampled by the previous grad student before me. We also see yellow points, which are the trees that are 0 to 200 years old, green, which are 200 to 400, and then blue, which are 400 to 600. You'll note that there's no dark purple points, which denote trees over 600 years old. And then here we have the low salinity site, which was where fieldwork supported by this award took place this semester. So here we see significantly more old growth trees seen in those dark purple points. We also see a lot of these white points, which are incomplete cores that didn't reach the center of the tree due to rot. This is really common in the old growth bald cypress trees, but it means we couldn't get an accurate age because the core did not go all the way to the center. This site is a healthy forested freshwater wetland, as you can see in the inset photo. And thanks to the help from this award, my team and I were able to take 76 new tree cores here this semester. Additionally, thanks to the help from this funding, we were able to do potential new site recon by boat at two potential new sites, Town Creek and the Northeast Cape Fear River. So at Town Creek, seen here, we found the ghost forest to be much more extensive than we were used to at Smith Creek due to its location closer to the mouth of the Cape Fear River. Old growth trees were much farther upriver than expected, seen in the map on the bottom here. And they were significantly more rotted than we were used to, which is why you see again those white points denoting the incomplete cores. The tree in the photo was definitely likely over 800 years old, but again, we couldn't get a complete core. But thanks to this trip, we also established an understanding of the salinity gradient using CTD castaway measurements, as well as a better understanding of the ecosystem here, which had an extensive salt marsh very far up this creek, as well as higher tree diversity than we were used to seeing. We also performed recon at the Northeast Cape Fear River itself, collecting sediment samples for foraminifera analysis. These are used to indicate when salt water has moved into an area, so you can get a better idea of how long the salt has been here and when it moved in. The trees that we were taking cores of here were also younger than the trees that we were used to at Smith Creek. But this site provides context for some direct response to the dredging, as all the trees that we cored were along the actual Northeast Cape Fear River rather than one of the tributaries like Smith or Town Creek. Further, this award funded necessary equipment for field and lab methods. So, for field work, we used funds for equipment including boots, field notebooks, safety equipment, a new GPS unit, and tagging materials for selective tree coring. In the lab, I mount the collected cores on specialized core mounts, and then I sand them down with a belt sander and a hand sander until they are both flat and have clear enough resolution to see the cells, which are then scanned on a flatbed scanner at a very high resolution. I then use a couple of dendrochronology programs to measure the rings and create a chronology of tree growth in the area. The Aaron K. Rice Award funded these lab materials as well, including core mounts, sandpaper, sanding safety equipment, and a specialized magnifying glass for reconstructing the cores before I mount them because they often fall apart. And then finally, over the past summer, I have been building a chronology of tree growth in the Northeast Cape Fear watershed over the past approximately 800 years, examining the relationship between those past suppression events that are driven by climate perturbations, such as drought potentially, trying to tease apart the impact from current anthropogenic drivers and sea level rise. In doing so, I'm hoping to establish whether current dredging related to suppression that we're seeing is unprecedented in that full 800 or so year dendrochronological record. And in looking at this figure again that I had brought up before, we see those dredge events and the suppression of tree growth in response. But if we were to shrink that down and then expand it all the way back further in time, we can expand the length of the chronology to look for those potential historical suppressions that I have kind of denoted in the gray dotted line. And likely those are, again, going to be driven by climate. And it's likely that they won't be to the extent that we're seeing now, because if they had, the trees wouldn't be here to tell this story. I wanted to say thank you again to the Rice family for this award and the opportunity to honor Aaron's legacy through the continued research in this field. Because of this funding, I was able to complete necessary field and lab research for my thesis, as well as fund parts of my tuition, which I would not have been able to do otherwise. And while I did not know him, it's very clear that Aaron's legacy in this department and in the field of geoscience as a whole is very vast and impressive. And he really embodied the kind of student and professional that I would like to be. And I'm honored to have been given this award. And I wanted to say thank you again for the opportunities that it's provided to me and my research. Thank you. And then finally, in speaking on tips for applying to grants to other students, I think it's really important to note that no grant is too small to apply to. Smaller pockets of money really add up and can be incredibly helpful for moving individual pieces of your research along. You should also tailor your responses to an institution's goals and their mission. Generally, many funding bodies will ask similar questions, so it can be kind of easy to fall into this pattern of answering them all the same. But by adjusting your responses and research to the goals of the funding body itself, even in just small ways, can really make your application stand out a bit. I would also say ensure that what you are requesting is within the scope and the requirements of what the institution will or won't fund. Many funding institutions won't pay for tuition or travel assistance, and you might have to find other sources for those. Especially a lot of those smaller pockets of money might not list those requirements on their website, and so you may have to reach out to somebody. So definitely don't be afraid to do that either. Additionally, it's likely that whoever is applying or reviewing your application is not going to be an expert in your exact field, and they're almost definitely not going to be an expert in your exact project. And so it's really important that you explain necessary terms and don't use too much unnecessary jargon or details, but really, find a balance between those two. I personally find it helpful to try and take myself out of the position of the person who's the expert on your project and try and explain it to myself as somebody who would have no prior knowledge beyond what the general scientific community would already have. And this is really important for like any writing and especially science communication in general that's going to be done in grad school and beyond. So this could provide like a great opportunity for practice. And in that same vein, be concise, be clear about your hypothesis, your methods, and if it's required for the grant application, your expected results. Really just don't make a reviewer have to search or infer what you're trying to say. That is all I have. I wanted to say thank you again to the Rice family for the support of this project and to please reach out if you have any questions at all. Thank you.